Let's take a look at this suggest why question, which asks us to use our own knowledge in this novel context. Use the information in figure 2.2 to suggest why naked mole rats do not feel pain when exposed to acid. We've been given a context here that naked mole rats are tolerant of pain caused by acid and information on their pain receptors, including a diagram, and we are going to have to propose an explanation for this, which will require us to use our own knowledge to interpret the information which we have been given. In OCR A Level Biology, we are not expected to know how pain receptors detect acid, let alone how those in naked mole rats work. However, we are expected to know that acid is caused by an abundance of hydrogen ion. Similarly, we may not know how pain receptors in naked mole rats work, let alone how they detect or maybe do not respond to acid, but we do know how pain receptors are generally supposed to operate. So let's begin by orientating ourselves to establish what we do know. We have the cell membrane here of a pain receptor in a mammal. It says that the first channel on the left is an acid-sensing sodium ion channel. This channel opens in the presence of hydrogen ions. So if they are opening in the presence of hydrogen ions, then that means they'll allow sodium ions in to the receptor, which will cause depolarization, which is the first step required to initiate an action potential. The second element on the cell membrane is that of a voltage-gated sodium ion channel. Now this should be a very familiar feature, because these are required to allow sodium ions in for depolarization proper to occur during an action potential if the threshold value is reached. So we've used our knowledge to identify how receptors are working within the context of this question's provided information. Now let's try and apply what we've identified over to the pain receptors in naked mole rats. So on the naked mole rat pain receptor, there is also an acid-sensing sodium ion channel. This channel opens in the presence of hydrogen ions, so the receptor detects stimuli and causes a depolarization. So the mechanism is the same as we currently know. However, in the voltage-gated sodium ion channel in the naked mole rat, this channel is prevented from opening by the presence of hydrogen ions, so this mechanism is different. Now this means that even if the threshold value is reached, the voltage-gated sodium ion channel will not open. So this means there's going to be less depolarization, which is going to prevent us from reaching the threshold value, so an action potential will not be triggered. And if there is no action potential, then there is no electrical impulse, and without an electrical impulse, there will be no information sent to the central nervous system to inform the naked mole rats of pain. So we have now identified our mechanism by which the naked mole rat is not feeling pain. So, because less voltage-gated sodium ion channels are opening in the presence of the hydrogen ions, that is resulting in less depolarization, so no action potential is being generated in response to the acid stimulus, so no information is being transmitted along the sensory neuron from the pain receptor to the central nervous system. The key takeaway here is that when you are confronted with a question presenting you with a novel context, you must first identify what you do know in terms of what the most similar mechanism or context that you have learnt about is, so you can transfer it over and retain the relevant information that you do know to work it out.